So I'm Mike Dustin, and I lead the cell dynamics platform at the Kennedy Institute of Rheumatology at the University of Oxford. It's my pleasure to welcome you to this online annual Oxford Imaging Symposium. To give a brief background, my co-organizer, Anjali Kasimbi, was kind of the driving force for starting this series back in 2018. So the Kennedy Institute Imaging Symposium in 2018 was followed by a larger departmental symposium in 2019 and an even larger Medical Sciences Division symposium with speakers from across Europe in January 2020. So we're rolling along really well with these in-person meetings. And then COVID-19 hit, and so we were unable to have an in-person symposium this year, unfortunately. But the pandemic, while really a catastrophe worldwide, has opened up windows on the world, through our computers. So today we have this online Oxford Imaging Symposium with speakers from around the world, literally. So in fact, our first speaker is staying up late in Australia, and our last speaker is going to be getting up very early in the US West Coast to be with us. So we're really grateful for all their efforts to join and make this a really interesting symposium. So our topic is a very exciting area, and we're looking forward to 5 talks on intravital microscopy in biomedicine, which will give us insights into the dynamic multicellular dynamics of multicellular life across a range of tissue environments and even up to entire developing embryos. So this is an area of deeper interest for a number of labs at the Kennedy Institute and across Oxford. So just some housekeeping issues. This is a standard Zoom meeting, so please keep yourself muted during the presentations, and we'll have a brief Q&A with each talk. So please type questions in the chat to everyone so we can all see the questions, and we'll basically try and get to as many questions as we can, either by reading them or letting people unmute and ask their questions. But I'm sure we won't have time to address all the questions. So there's a hashtag in Twitter, hash Oxford Intravital, one word, that you can basically attach to your tweets, and then you can basically use that as a search term, and we can have like an asynchronous discussion on topics related to the symposium, and hopefully some of the speakers will check that out and continue the discussion online that we've started here today with the symposium. So we're going to record some of the talks, so you'll see like our little record button on, and then this is what the speakers, as is with the speakers agreement, and so just wanted to let everyone know that. So the questions and things may also be recorded and part of a record that basically can be accessed by registered attendees in some cases online publicly. So I'd now like to hand over to my co-organizer, Anjali Kasimbi, to introduce the first speaker.
Hi. So yeah, good afternoon, everyone. So it's my great pleasure to introduce Tri Phan, who is a group leader at the Garvan Institute of Medical Sciences in Australia. And as Mike said, he's giving this talk at around midnight today. And he basically in 2010 established the Intravital facility, the 2-photon facility at the Garvan Institute. He has been working on the in vivo B cell responses in the imaging the in vivo B cell responses in the lymph nodes. And recently, I think so what he is going to present is the work which he did on the osteoclast, the osteomorph identification of the new cell types. And this is in close collaboration with the Bone Lab of Peter Croucher and with Michelle McDonald's. And yeah, this is also something my lab and my interest is close to that, the bone marrow microenvironment. And we are very looking forward to your talk. And thank you so much, Tri, doing this at this time during the midnight. It's to you.
Great, thanks very much. Can everyone hear me?
Yeah.
It's Tri, we can hear you.
Yeah, okay, awesome. All right, so thanks very much, Anjali, and thank you, Mike, for giving us this opportunity to share some of our recent work, which is now published. But, I always love an opportunity to present this data because it's obviously very exciting for us get feedback and to find out, where to go from here. So I'm just going to take you back a little bit and pay a little bit homage to Uli von Andrian, who developed really what has been the go-to method for intravital imaging of bone until recently. And that was a method that involved imaging of bone in the skull, taking advantage of an optical window in the skull where the fontanelles meet at this position here called the Bregma. And of course, David Scadden with Christina Lo-Kelso famously applied that method to image hematopoietic stem cells in the bone. But of course, the skull is a non-weight-bearing flat bone and is derived from intramembranous ossification. And working with Peter Croucher, we became much more interested in weight-bearing bone, which is derived from endochondrial ossification. So we developed a new method for intravital imaging, and it takes advantage of the fact that when you work in mice and you peel back the skin over the leg, it becomes apparent that as soon as you do that, you can actually see the marrow inside the tibia. And that to us indicated that the tibia was actually optically at least suitable and perhaps transparent enough to be perceived with a 2-photon excitation laser. So we developed this method for intravital imaging of bone in the tibia, and a few years ago we used it to study dormant multiple myeloma cells. And in this video here, you can see the myeloma cells arriving in the bone, in the tibia, and then colonizing the endosteal bone surface. So what we wanted to do then was to use this method for imaging to then image osteoclast formation, function, and fate. And to do that, we really needed a high fidelity system for in vivo tracking of osteoclasts. And we took advantage of the fact that osteoclasts are formed from the fusion of macrophages. And so we thought that if we had a system with which we had a red macrophage and a green macrophage, then the osteoclast in that system would then be formed by the fusion of those two cells and would express both a red and green fluorescent reporters. So a double labeling system. So we took advantage of that and developed a mixed bone marrow chimera system. And typically we would use a Lysem-Cree TD tomato donor and mix it with a mafia donor. So we have green and red lineage macrophage cells, and use that to reconstitute the bone marrow of lethally irradiated mice. And so in this system, then the osteoclasts that were generated would be double labeled red and green. And for us, that was very advantageous because in our initial forays into imaging osteoclasts, we were quite frustrated by the fact that a lot of the reporter systems that we used were quite leaky. So in this system, what you can see here that is that double label cells, so cells that are expressing both Lysem and CSF1 receptor, are in fact large multinucleated cells. We find them residing on the endosteal bone surface, which is what you would expect for an osteoclast. And they also resorb bone, and we show that by showing that they take up bisphosphonates. They also express active cathepsin K enzyme. And what was really interesting was that instead of the large rounded cells that we were seeing in textbooks, what we saw was a large cell that had a lot of dendritic processes that was forming an interconnected network across the endosteal bone surface. Okay, so what does it look like? So in a lot of the movies I'm going to show you, we're just going to concentrate on the red fluorescent signal just so we can concentrate on the biology. But what we can see here is that you have this large osteoclast which has all these processes, oops, I may have stopped the movie. But when you track the process and you track the cell, actually they're relatively very stable over long periods of time. So in the steady state, what we see is that these osteoclasts are largely non-motile cells that are extending these processes that may connect and allow them to interact with other neighboring cells, including other osteoclasts. So what happens if we perturb this system? So what we did was we injected soluble rancor, which is a TNF family member that is a very potent stimulus for osteoclast formation and function, and a very strong driver of osteoclastic bone resorption. And what you can see in this 2-hour movie is that as the rankle goes in, the osteoclast retracts its processes, becomes more rounded, and they actually start migrating towards each other. And what was really interesting was that although these cells were already large multinucleated cells, they were actually fusing to form even larger polykaryons. And you can see this here with the tracking where we've color-coded the individual osteoclasts to show the retraction of the processes as the cells move towards each other, and then eventually fusing with each other to form a much larger cell. And I'll just let the movie play through. And of course, you know, cell-cell fusion is not something that's entirely new. I guess we were a little bit surprised that the osteoclast would do this. But what happened was that when we watched the osteoclast for a little bit longer, okay, so I'm just going to show some Z stacks here to show that these cells have in fact fused, and then a 3D volume to show you that as you rotate through that these cells have in fact fused, and it's just, you know, not two cells overlying each other on different Z planes. But what was really interesting is that when we watch the movies for a bit longer, not only did we see cell fusion, but in fact, sometimes we were seeing the osteoclasts breaking up into smaller cells. Now, these smaller cells actually were in fact still motile and still able to migrate. And this was really interesting because now what we're seeing is not just osteoclastic cell fusion, but osteoclastic cell fission. So again, here we've mapped it just to make it easier to see. So you see this large osteoclast here, pulling back, separating from this osteoclast. And then breaking up again. Into smaller units, which were still viable and motile. So these smaller cells were really curious for us because up till now, the dogma was that osteoclasts undergo apoptosis, and so they were considered short-lived. But of course, really, really nice work a couple of years ago from Frederick Geismann's lab using parabiosis showed that, in fact, osteoclasts were much longer lived than we had realized. And of course, a lot of the textbooks and a lot of our knowledge about osteoclasts have derived from in vitro culture systems. And it may well be that, in fact, in vivo, what we're seeing is entirely different. And in fact, that's obviously why we're all here and why we're all sold on the power of intravital imaging. So the current dogma is that osteoclasts form from these precursors, and then once they finish their job, they undergo apoptosis and die. So one of the things then that we wanted to make sure was that what we were seeing was not osteoclastic apoptosis. And so to actually see what apoptosis looked like in vivo, we actually used the 2-photon laser to induce apoptotic cell death and image that and then compare that with this fission that we were seeing. And you can see here that after a localized laser injury, the osteoclasts would break up, and instead of breaking up into large cells that were still motile, they were breaking up into small subcellular fragments. And instead of being motile, these fragments are actually non-motile. And at the same time, what we're seeing was a large influx of macrophages, which were cleaning up this apoptotic cell debris. And over time, you get loss of fluorescence, and the osteoclast is cleaned away and disappears entirely. And in fact, when you put them side by side, what you see here when you track the cells with fission, as the cells fission, they still retain a cell volume. So at the end of the imaging session, we still see cells with volume, whereas with apoptosis, the fluorescence signal disappears entirely, and there's no volume at the end of the imaging session. And at the same time, you get this influx of macrophages interacting with the apoptotic cell fragments and clearing it away. So that made us wonder, well, actually, what's going on here? What are these smaller cells, and are they actually a new cell type? So what we did was ask, what's the fate of these smaller cells? And so here in a 7-hour movie, which is if we watched it for a much longer period of time, you can see all these fission events occurring. But the curious thing was that as we watched these cells breaking up, sometimes what we were seeing was that the cells would actually come back and fuse again. So what we're seeing was the osteoclasts were fissioning into smaller daughter cells, and these daughter cells would then migrate and then fuse back into larger osteoclasts again. And that suggested to us that, that there was a cycle going on here, fission and fusion, and that instead of undergoing apoptosis, that osteoclasts were actually fissioning into a new cell state. And hopefully the movie is a bit slow. We'll show you a bit of the cell tracking. But anyway, so what we thought was, okay, we're seeing red macrophages and green macrophages forming osteoclasts, and they're breaking up again. And that reminded a lot, actually, of the Mighty Morphin Power Rangers. And, here's a red and green Power Ranger. And so we thought, why not? Let's just call them osteomorphs, because that's what they reminded us of. And so we came up with this new model that instead of undergoing apoptosis and dying, osteoclasts were actually fissioning into these osteomorphs and then recycling back into an osteoclast. So really, are these cells for real? Can we find any orthogonal evidence for the existence of these osteomorphs? And so we took advantage of the fact that if they're fluorescent, we might be able to isolate these cells. And in fact, here you can see when we performed flow cytometry, we can identify cells that were both red and green in the mixed bone marrow chimeras. And when we looked at the fluorescent signal for the fluorescent bisphosphonate zoledronic acid, that the only cells that have taken up the bisphosphonate are the double labeled red and green cells, suggesting that the double labeled cells had accumulated zoledronic acid, and in fact suggesting then that they would derive from an osteoclast that had resorbed bone to have accumulated that zoledronic acid. So taking advantage of that fact that we could fax these cells, we then sorted them, and then both through in vitro cultures and then ex vivo on Calvaria, we could show that these triple positive cells, in fact, so they had red and green signal and had fluorescent bisphosphonate, that they could in fact fuse again and form osteoclasts on Calvaria, indicating that they had the potential to form osteoclasts. But what was really important for us then is that this gave us a mechanism to then do single cell sorting and ask, and single cell RNA sequencing and ask, well, are these cells in fact bona fide separate cells? And so when we do this, when we sorted the single positive cells and the triple positive cells from both the bone marrow and from the bone, and our hypothesis here was that the osteoclasts would be on the bone surface and the osteomorphs, on the other hand, because they'd broken off and were much more motile, would also be found in the bone marrow cavity. But so when we did that, what we found was that despite sorting these different populations, the cells actually clustered very nicely, and they clustered into these 3 groups, which you can see here. When we analyzed it, it seemed that the clusters were largely triple positive cells that were found on the bone surface, which we thought most likely corresponded to the osteoclasts. Single positive cells, whether they were on the bone surface or in the bone marrow space, clustered together, and these we just called macrophages here. And then this group in between, which were actually largely triple positive cells in the marrow, and we thought that these were most likely to be the osteomorphs. So we have 3 clearly distinct transcriptional states, and suggesting to us that perhaps the osteomorphs were indeed a new cell type. What was really interesting was that this cell population, the osteomorphs, they expressed a lot of genes when you looked at the upregulated genes that were shared with the osteoclasts. And you can see here that 430 of the genes that were upregulated in osteomorphs compared to the macrophages were also expressed by osteoclasts. And these included what could be considered canonical osteoclast genes like NFAT-C1, RANK, CSF1 receptor, TRAP, cathepsin K, and the vacuolate ATPases that are required for the acidification during the bone resorption stage. So what was really important for us was to establish whether, in fact, these osteomorph upregulated genes were important in bone development. And so we were very lucky to collaborate with Graham Williams and Duncan Bassett, who's been working with Peter Croucher on the Origins of Bone and Cartilage Disease program, which is funded out of the Wellcome Trust. And they had generated a large number of knockout mouse, and we, looking in their database, we found that 40 of the osteomorph upregulated genes had in fact been knocked out by this OBCD program. And 17 of those 40 knockouts actually turned out to have a bone phenotype. And so on the right here, I've shown as 3 examples, DDX56, MYO7A, and WDR89. So these are genes that either in the heterozygous or homozygous state when knocked out have bone structural and functional effects. So they either cause loss in bone or gain in bone content or bone strength. What was really interesting, though, is that of the 17 genes that we found were involved in bone structure and function, 11 of them had actually not been described before, and 2 of them were actually uniquely expressed or uniquely upregulated, rather, in the osteomorphs, and were not upregulated in the osteoclasts, suggesting that this is a cell type that's going to give rise to really interesting biology down the track. So, of course, this is in mice, and so we looked at of the 581 upregulated osteomorph genes. We found that 520 of them had human homologs. And in fact, 22 of these genes are associated with monogenic skeletal dysplasia. So indicating that these genes are actually important in bone disease in humans. And we were lucky enough to collaborate with John Kemp, who was able to look in the UK Biobank study where they looked at estimated bone mineral density and found that 71 of the 520 genes actually significantly associated with changes in estimated bone mineral density. So this suggests that these genes that are upregulated in osteomorphs are actually quite important, not only in monogenic bone diseases, but also in polygenic diseases such as osteoporosis. So I'm going to finish there by summarizing what I think we've done. And so, our lab actually concentrates on doing intravital microscopy as a mechanism to discovering new biology, and then we combine that with gene expression. I think this project really gives a great example of how that workflow pans out. So what we've shown, I think, is that by intravital imaging of osteoclast dynamics, we've discovered a new cell fate, and that the osteoclasts, instead of undergoing apoptosis, they recycle, and they recycle by this new cell type that we've called osteomorphs, and that the genes expressed by these that are upregulated in these osteomorphs are actually very important in skeletal structure and function, not just in mice, but also in humans where they're implicated in human diseases. Just in the interest of time, I didn't show you some of the data, but one of the things that we're able to model in our mixed bone marrow chimera system was what happens if you withdraw from Denosumab. So Denosumab is a very potent inhibitor of rank ligand signaling, and it's a blockbuster drug that's used to treat osteoporosis. But one of the things that's been discovered is that when you stop Denosumab, patients have these paradoxical rebound vertebral fractures. So we were able to inject OPG-FC in mice to mimic the effect of Denosumab, and after administering OPG-FC for weeks and withdrawing the drug, we see the same effect. We see accelerated osteoclastic bone resorption, and the data I haven't showed you, it's due to the accumulation of the osteomorphs, and so that when the OPG-FC is washed out, the osteomorphs are actually primed and ready to reform rapidly osteoclasts, which then very actively resorb bone. So I'm going to finish there by just thanking Peter Croucher, who's just been an incredible collaborator. And I think this project also is a really good example of the value of interdisciplinary research, because Peter is a bone biologist and I'm an immunologist. And of course, Michelle McDonald, who did all the intravital microscopy, and Wang Waku, who did all the single cell RNA sequencing. And of course, this was not possible without the support and collaborations from literally a cast of thousands. And particularly, I just want to call out Graham Williams and Duncan Bassett at Imperial College and John Kemp for their wonderful assistance right at the end, when we really wanted to see what these cells were actually doing, and of course our funding support. And I'm going to stop there and hopefully be able to answer some questions.
Thank you, Tri, for this great talk and this amazing work. So there is a question in chat from Mike. Do you think that osteomorphs help maintain the turnover of the bone or the bone marrow niches in the bone marrow? Like, do they interact with the other bone cells?
Yeah, no, I think that's a great question. So you know, I think in the steady state, you know, we see osteomorphs in the steady state. One of the things I didn't mention is that we can see double labeled cells in the blood. And so we think that these osteomorphs are actually able to migrate and recirculate. And in fact, one of the really interesting questions is whether what people have called osteoclast precursors in the past may well be these osteomorphs. We don't have evidence directly that they are in fact involved in the turnover of bone spicules, but definitely one of the areas that we're really actively exploring is whether osteomorph, you know, this dynamic, this equilibrium between osteoclast and the osteomorph state is important in regulating bone turnover, particularly in disease states where we think bone turnover is extremely important, not just osteoporosis, but for example, in the reactivation of cancer cells in the dormant cancer cell niche in bone.
Yeah, so you may, so with this you mentioned like the osteoclast, people previously thought this as an osteoclast precursors, but then the osteoclast precursors, what people have reported is that they are there only in the young bones, like they basically decline. At least the osteoclast precursors are known not to be there during the aging. So what is it the same for the osteomorphs or what?
Yeah, no, I think that's a really good point. And I think, you know, I go back to that really beautiful work from Frederick Geisman's lab a couple of years ago, where they did parabiosis and showed that, you know, osteoclasts are much more long-lived. One of the things that I think is going to be really interesting, as you point out, is what happens to these cells in the aging process. You know, how do they change with age, and whether that impacts on osteoporosis?
I mean, do you have any questions for Kat? No. Okay. No. Yeah. So I have a question. If there is any question, please drop it in the chat. And if you want, you can even unmute and ask, but it might be better to have it on the chat. So I have a question like where, so, you know, these osteoclasts are, so when you image in the cortical bone, so basically what you, with your imaging setup, you are only imaging the cortical bone, not the trabecular, I guess.
That's, we're predominantly imaging, yes, cortical bone. There is a, you know, there's a really nice optical, a natural optical window in the bone where it's actually thinnest in the tibia that we tend to gravitate towards because that's where we can go deepest. We can also image up towards the growth plate in the tibia, but we don't, you know, don't go that deep at the moment. But we've, you know, one of the things that we're really working on is trying to implement adaptive optics in our 2-photon microscopes so we can actually go deeper.
Okay, so like, so you were imaging the cortical bone. So if you go within the sections, like, do you have any cell surface markers for it which distinguish it from the macrophage? I mean, can they be spatially mapped in the bone? Because if are there any surface markers which distinguish the osteomorphs versus the macrophages and the osteoclasts?
Yeah, no, I think that's kind of like one of our ambitions. So far, at the moment, we've got a number of markers that we can find expressed by the osteomorphs. Unfortunately, a lot of them expressed by other cells, including macrophages. So at the moment, by flow cytometry, you can use gating and use multiple cell surface markers to distinguish them. But really what we would really like to do is to have a single or two markers, ideally, that would allow us to find them. And really what down the track, what we'd really want to be able to do is to find them in people.
Yeah, because if there are cell surface markers, other thing like the osteoclast and the blood vessels closely interact.